This is Biochemistry Unfolded, from atoms to algorithms, a weekly podcast on the topic of computational biochemistry and biophysics. Well, good afternoon, and welcome to the fourth episode of my weekly or semi-weekly, I'm not sure exactly if I have a schedule yet, of my podcast, Biochemistry Unfolded, from atoms to algorithms. And this is pretty exciting. Actually, in the past week, if you're watching this on YouTube, the total number of subscribers on YouTube hit over 100 subscribers, and it keeps growing, so that's pretty exciting. This is great news because that means people are actually listening to some of the things I'm saying, and there's some other videos in my YouTube channel. So I think if you haven't seen some of those videos, posted a really interesting video in the last week on caffeine, and so that's pretty interesting. And there's some stuff about the sympathetic and parasympathetic nervous systems. It's great interacting with people. If you have any comments that you'd like to share, any burning questions you would like answered, please let me know, and we can certainly try and address that in a future episode. As far as episode 4, so this week I actually found a really interesting paper, and this paper was actually published just recently. So this is hot off the presses, and so this is in Nature, Structural and Molecular Biology. This is an article entitled Structures of Aberrant Spliceosome Intermediates on Their Way to Disassembly, published just before Christmas, December 19th, 2024. It was actually published online on January 20th, 2025, so fairly recently. The work comes to us from Kamal Soni. I'm not sure how you pronounce that. I hope I didn't massacre your name. There's several coworkers on here as well. And these are all at the University of Heidelberg in Germany, Heidelberg University Biochemistry Center, BZH. And so what they're talking about is the spliceosome. So what exactly is a spliceosome? The spliceosome, well, let's start with the central dogma of molecular biology. And I always, whenever I teach biochemistry, it's always a question in my biochemistry exam: what is the central dogma of molecular biology? So the central dogma of molecular biology is that DNA, which is the primary genetic molecule, that is transcribed and undergoes a process of transcription in the nucleus to messenger RNA. And so DNA is double-stranded, messenger RNA is single-stranded, and then that undergoes the property of translation at the site of the ribosome, which is usually in the cytosol of the cell, or it may be bound to the endoplasmic reticulum. And so then that's how proteins are made and proteins are constructed. Now, in a cell such as a human cell, there's probably 20,000 plus genes, maybe 22 or 23,000. I don't remember the exact number, but it's a lot. There's also a lot of introns and exons. And so there's actual gene material, and then there's non-coding DNA. So during the process of transcription, you need to transcribe what is important to make the proteins you need at any given time. You don't necessarily need every single thing in the genome all the time. It's kind of like a program, right? And it's kind of like running a Microsoft Word piece of software. Microsoft Word is probably the foremost software that most people write papers on and use for their word processing. There's a lot of functions in Word. You can bold, you can underline, you can change things like the font and the font characteristics. There's also things like adding tables, adding images. There's a lot of code within Microsoft Word, just like there's a lot of code within DNA. Now, whenever you're running Microsoft Word, let's say you're just editing a simple document, you don't need a table. You're not going to add an image. Well, you don't need that code that does that. You just need other code that runs Microsoft Word. You may not use the spelling checker. Actually, you probably should, but that's beside the point. DNA works the same way, right? When you're running a program, you don't need every single thing that's in the code. You just need the specific functions that you're using at any given time. Whenever you're transcribing a gene, you don't need the entire code in the gene at any given time. You just need a small segment of that gene. And so the way Mother Nature accomplishes this is by splicing out code that is non-coding, and then you get the code that you need, right? And so the spliceosome is a complex that accomplishes that. Another way to think about it is when we edit YouTube videos, we're going to use a lot of different videos, maybe different audio tracks, the music for Biochemistry Unfolded. I add that in. I add the intro, I add the outro to that, and I splice it together the same way we splice together genetic code, right? So the spliceosome is a small complex. It's a ribonuclear protein, and that's responsible for that pre-mRNA splicing. And occasionally errors take place. Occasionally we make errors in YouTube videos as well too. That's really easy to do. And so what's particularly notable about this particular study, now there's several, I think in 2015 or so was the first, the structure of the spliceosome was actually published. So that's, that makes this a little bit less notable, but what makes this notable is that this is actually an aberrant spliceosome. What does that mean? An aberrant spliceosome means that this is actually not functioning properly, right? This is malfunctioning, right? And so that makes this notable because we can understand how the spliceosome works and how the spliceosome malfunctions. We can understand how it malfunctions in specific diseases such as cancer or neurodegenerative diseases such as Alzheimer's or Parkinson's disease, for example. The other notable thing about this particular paper here is that they obtained the structure via cryo-electron microscopy. And so what exactly is cryo-electron microscopy or cryo-EM? That's a fairly new thing. So, the gold standard in protein structure determination is X-ray crystallography. And that's where you grow crystals of your protein. And then you put those crystals onto a little pedestal, you zap X-rays at it, and you're able to obtain a diffraction pattern, which can be correlated with the electron density of the protein. And hence you can see, if you can see where the electrons are, you can see where the atoms are and put together the protein that way. Now that requires a crystal. And it's fairly difficult to grow crystals of protein. It's also not necessarily natural, but it's essential in X-ray crystallography because it's essential for all of the atoms to be oriented the same way, the same direction, or else you're not going to get diffraction. You're not going to be able to look at the electron density, and hence you won't get the structure of it. So cryo-EM, how does that differ? So cryo probably indicates low temperature, of course. Not probably, it does. And so the sample, your sample of your protein or whatever your protein complex, as the case may be here, the case is is your sample is rapidly frozen in this thin layer of ice, and then we use an electron microscope. So instead of X-rays, we're using electrons, which have a smaller wavelength than the X-ray, and we shoot electrons at that frozen sample, and then we can reconstruct a 3D model of these images. So it's kind of like that's why it's called Cryo-Electron Microscopy. It's kind of like a microscope, except instead of using light, we're using electrons right there. And so this technology can obtain near atomic resolution. So we're talking 2 to 3 angstroms. And the lower that is, the higher the resolution, right? And what's also cool about this is, since we're not in crystals, we are frozen, but we're not in crystals, but we can handle fairly large complexes, a little bit larger than what X-ray crystallography can handle. And we can also handle some dynamic complexes and things that more closely resemble what takes place in nature. So something like a spliceosome, for example, we can look at how the spliceosome is malfunctioning, and that's also very important as well. And so we don't have to crystallize the protein. That's kind of important right there. And so they used Cryo-EM to capture the snapshots of a spliceosome in the bacterial intermediates from the bacteria. The bacterium is Schizosaccharomyces pombe. And so that's the bacterium of study right there. Now, what they found in this study is they've identified a key DEH box helicase protein. That's GIH35. And that's a G-patch protein pair, and that maintains catalytic dormancy, right? Now, what does that mean, right? That's kind of interesting. Now, in a normal spliceosome, helicases facilitate those splicing reactions by promoting conformational changes. But in the spBd complex of the spliceosome, those structural changes at the 5' splice site, they lead to an extranucleotide insertion. And so this locks the active site in a non-functional state. And then the presence of this GIH35 at the spliceosome surface, this further prevents the regression to catalysis. So that's what we mean by it maintains that catalytic dormancy. So it kind of prevents things from further progressing, right? And so we have structural remodeling of the active site, and this leads to the insertion at the 5' splice site, which triggers recruitment of another protein complex, the Ntr1 complex, and that mediates disassembly, right? So what does this all mean? Let's just use the words quality control, right? And so this is essentially engaging in the quality control mechanisms of splicing, so that we actually have something that actually, the final transcribed mRNA sequence that's spliced together, that's going to go to the ribosome, that's going to be accurate. When things don't work, then the complex should fall apart, and then we don't get the mRNA, right? So that's essentially what's happening. So we used Cryo-EM to get that structure. They also used protein purification and affinity tagging. And so what is that? So obviously you need to obtain, if you're going to get a good crystal structure, you've got to get the protein purified in such a way. And so there's a number of ways we can purify proteins. In this case, we're using affinity tagging. So this employs a split tag approach. And so we have little affinity tags, which have complementary structures to parts of the protein. And so then they kind of bind to it, and then they carry it, and they're able to purify the protein by binding to specific parts of the protein. In this case, the split tags are 2 different key proteins, Ntr1 and PRP43. And these are both involved in the disassembly of the spliceosome. So that's kind of how we actually purified the protein. What they also used, in this paper, they used mass spectrometry, MS. And so mass spectrometry is a way we can actually look at protein structure and identify specific things about the protein sequence. In this case, they used crosslinking mass spectrometry to identify protein-protein interactions within the spliceosome. And that's important because when we identify specific protein-protein interactions, we know that one protein makes contact with another protein at these points, and another protein chain makes complex with another part of the protein at a different point. And so that's all interesting. So by chemically cross-linking the proteins that are in close proximity, we stabilize the interactions before the complex is analyzed by mass spec, and then we identify those cross-linked peptides so the researchers could determine which proteins are physically interacting. And that's important because, we've got a lot of pieces to put together. This particular protein complex is many proteins, not just 1, 2, or 3. And it's a very complex protein, and it's very dynamic because everything is also in motion. So by cross-linking these peptides by the mass spec, we're actually able to obtain the structure, and then we can validate that structure by looking at specific protein-protein contacts. So that's very important. And then we looked at the RNA sequencing with RNA Seq. And so this analyzes the pre-mRNA substrates that are targeted for discarding. And they found, what this found is they found that certain transcripts, such as RPL39, exhibit a specific sequence motif that causes malfunction in the spliceosome. So those are the major methods that they used in this paper. So what did they find? What are the main results? First of all, they found that the spBd complexes resemble an intermediate between B and intron lariat spliceosome states. So they found that the structure kind of resembles those intermediates. And they found that the malfunctioning stabilization of the U5 loop with the 5' exon leads to an extra nucleotide insertion, and this disrupts its proper splicing, right? They also looked very closely at the role of the GIH35 Gpl1 pair, and so Gpl1 kind of acts as an anchor for GIH35, so they're going to interact closely together, and then that binds very deeply in the active site. The interaction of both of these proteins together prevents branch point docking, and that's how it maintains catalytic dormancy. So that's kind of how it shuts it down to stop things from happening, right? Because GIH35 is key in that catalytic dormancy, right? As we stated before. Now the Ntr1 complex is what actually mediates the disassembly coming apart, right, of this entire complex. And so the spliceosome recruits the Ntr1 complex from several other complexes. That's Ntr1, Ntr2, PRP43, and CWC23. So altogether, all these proteins maintain that quality control element when things go bad, right? That's kind of one of the key aspects of this particular paper. And so we identify the mechanism by which splicing errors are recognized and then discarded by the mRNA maturing. And then we have specific sequence motifs, quadruple adenine motifs in the 5' axon. These are associated with splicing errors, right? So essentially, quality control, when you're doing programming, for example, if you're programming Microsoft Office, right? Well, do you know how much code is discarded when you code? Yeah, you go through a process of debugging, right? Things don't always work right. I'm actually debugging a Python script right now to analyze, do different things with Gaussian. And yeah, it's kind of a pain. We go through different iterations of code. Sometimes it works. Most of the time it doesn't. We discard the code, right? We need mechanisms to discard our computer code the same way when things go wrong with Mother Nature. Yeah, we need to discard that code as well too. So that's the quality control step is an integral part of protein coding, right? So that's very important. Now let's look at the figures in this paper here. Let's look at figure 1, right? And trying to think where I can put figure 1 on here. I think I'll put it maybe up here, right? I think that'll probably work. And so figure 1, this presents the Cryo-Electron Microscopy structures of 2 of our malfunctioning spliceosome intermediates. That's spBd-I and spBd-II. I think they're both on opposite sides here. And so these are stalled on their way to disassembly. So what the structures reveal, they reveal some insights into the molecular composition and organization of these spliceosomes, right? So the 2 structures are similar overall, but they show some compositional differences. In spBd-I, the Ntr1 complex is weakly present, and in spBd-II, the Ntr1 complex is more stably bound, and we can actually see that in this particular figure right here, right? Number 2 is we've identified the GIH35 Gpl1 complex. That's the helicase and the G-patch protein, right? And so these are clearly localized in both of the structures. The Gpl1 anchors to GIH35 to the spliceosome, and then it is allowed to influence the RNA remodeling and prevent further splicing. Number 3, the structural remodeling of the active site is visible, and so we can see the active site of the spliceosome in SBD complexes, and that deviates from our normal splicing states, right? And the 5' splice site exhibits a single nucleotide insertion. This causes excessive stabilization of the U5 loop with its interactions with the exon, right? And then we can also see a recruitment of the Ntr1 complex, which is preparing for disassembly of the whole thing, right? So that Ntr1 complex has Ntr1, which is a small peptide, and Ntr2. We also have PRP43 and CWC23. These are all little peptides, all little parts of the protein, and these are all involved in dismantling and taking apart the entire spliceosome that's malfunctioning, right? And so then the presence of SAF4 and BIS1, that's in spBd-II, that further stabilizes the Ntr1 complex. And that that's an indication that this whole thing is in the process of coming apart and being disassembled, right? So that's what we mainly see in figure 1 here. That is the visualization of that molecular basis of spliceosome quality control, right? So that's important. This is one of the first high-resolution structures of this spliceosome in disassembly state, preparing to come apart because it's malfunctioning. We've already had spliceosomes. We can see those. So this is one of the first ones. So we can see the quality control steps very clearly here. Now, figure 2, that's the next part. Let's look at figure 2 here. And so in figure 2, this shows the stabilization of the Ntr1 complex in the spBd-II state. And so this focuses on how the Ntr1 complex is recruited and stabilized in that spliceosome intermediate that is primed for disassembly. So what we're showing here is we see spBd-1 and 2 with particular attention to how the Ntr1 complex is becoming more stably bound to spBd-II, right? And so what is the significance here? We see structural evidence for how the Ntr1 complex is recruited and stabilized before disassembly. Number 2, we see the differences in spBd-1 and 2 that suggest a more advanced stage of the discard pathway where the disassembly factors are firmly in place. And number 3, we see the presence of another protein pair, SAF4 and BIS1, that suggests these proteins actually act as stabilizers that ensure the disassembly machinery is effectively recruited. So that again maintains our quality control in the overall process, right? All right, so what's the overall takeaway message here? What we're showing in figure 2 is that we're explaining how the Ntr1 complex transitions from fairly weakly bound in the spBd-1 stage to a stably bound spBd-II. This shows us how the defective spliceosome is to be disassembled. And so this is a very crucial step in splicing quality control. This ensures that those malfunctioning spliceosomes don't continue the splicing process and are in fact targeted for being broken down, right? So then if we go to figure 3, let's look at figure 3 here. That shows the interactions between Gp1 and GIH35 with the spliceosome. And so figure 3 shows how the G-patch protein, Gp1, anchors the helicase, that's GIH35, to the spliceosome. This interacts with key components of the whole spliceosome complex. And so Gpl1 protein, that's one protein that bridges with GIH35, and then that also bridges with PRP8. That positions the helicase to the periphery of the spliceosome, and then that reaches fairly deep into the active site. What we show here is how the GPL1 recruits GIH35 into that spliceosome. This ensures that the whole helicase is positioned correctly for the disassembly process. This also shows that GIH35 doesn't actually remodel the RNA in this state that's held in place by the GPL1 protein. And so this provides structural evidence for how that spliceosome identifies our defective complexes and marking them for discarding through the interaction between GPL1, PRP8, and GIH35. So we're kind of looking at everything, right? Now, what's also important, so the takeaway message here is that the protein GPL1 component, this acts both as a recruiter and an inhibitor, right? And so this anchors GIH35 while also locking that active site into an inactive conformation. And this is a key step in the spliceosome quality control pathway. What happens here is this ensures that the defective splicing intermediates don't proceed to catalysis and in fact are marked for disassembly. So that leads us to figure 4 in this paper. Figure 4 shows the GIH35 GPL1 complex as a helicase and a G-patch protein pair. So we're focused on the individual molecular interactions between GIH35 helicase and the Gpl1 GPatch proteins. So these are kind of coming together. And it shows how Gpl1 tethers GIH35 to the spliceosome, right? And so this interaction is actually crucial in stabilizing that defective spBd complex. That prevents your splicing progression, and that essentially primes the whole thing to be disassembled, right? So figure 4, this confirms the GIH35 and Gpl1 function as a helicase GPatch pair, right? And that's similar to the known splicing helicases such as PRP2 and spBd-II as well. It also shows that GIH35 is recruited to the spliceosome indirectly via Gpl1 instead of binding to the RNA directly. So the GIH35 doesn't promote splicing progression. It's in a dormant state, then it's ready for discarding. And so we have specificity of the GIH35 and Gpl1 protein interactions that support the model for these helicases overall. So what we get out of figure 4, this figure, this provides structural and functional evidence that the GIH35 helicase is not actively remodeling RNA in the spBd complex, but it's instead just held in place by the Gpl1, right? So it's acting as a recruiter, right? This supports the idea that the spliceosome intermediate's not progressing towards catalysis, hence the dormant catalysis, catalytic dormancy, but it's instead marked for disassembly. So it's not going to undergo its intended purpose. It's going to shut down, right? Hence its use as a splicing quality control mechanism, right? So overall, the study provides structural evidence of an aberrant spliceosome, hence the title, of an intermediate that is committed to final disassembly because it's malfunctioning. The findings suggest we have abnormal base pairing interactions in the 5' splice site that can alter the active site conformation and leading ultimately to premature spliceosome discarding, right? We look very closely, the paper looks very closely at the interaction between the GIH35 Gpl1 helicase GPatch pair. This plays that critical role in recognizing defective spliceosomes and preparing them for final disassembly. So what we've learned from all this stuff, hopefully, is how these proteins, how all these multitude of proteins, and I think I've mentioned a lot of little combinations of letters and numbers, and I hope I haven't confused everybody, but we've learned how all these proteins they've enhanced our understanding of quality control mechanisms, right? And all these quality control mechanisms ultimately prevent errors in the processing of our mRNA, right? Now, what is this going to lead to? What are the main implications for science? So, number one is we can understand splicing disorders a lot. We have a much better understanding of how the spliceosome works in genetic diseases, such as cancer, Alzheimer's, Parkinson's disease, other neurodegenerative disorders. And so these insights could help lead to potential therapeutic interventions. This could also lead to RNA-based therapeutic drugs or therapeutics overall. It could be a biological drug. We could use antisense oligonucleotides or small molecule therapies that target misspliced RNAs, right? We've also advanced our knowledge of Cryo-EM as well as RNA sequencing in uncovering a lot of complex interplay between the the interactions between the RNA and protein and their interactions thereof, right? So Cryo-EM, we're going to see a lot more Cryo-EM, Cryo-Electron Microscopy. That's going to be a huge advance coming forward. This could also lead to other advances in synthetic biology and gene editing. For example, understanding how this spliceosome identifies defective substrates will help improve CRISPR-based RNA editing. CRISPR, clustered regularly interspaced short palindromic repeats. That won, I believe, the 2020 Nobel Prize in Chemistry with Jennifer Doudna and Emmanuel Charpentier. So we can use CRISPR as a therapeutic method for modifying genes, and so that could be useful. And we could also look at alternative splicing regulation in gene therapy. So again, this has an implication that could be coming into play in like CRISPR development as well. So what's going to come next? There's a lot of different things we could look at this as a next step, expanding our insights into spliceosome quality control, right? Because now we've got knowledge of the spliceosome that's actually defective and how it disassembles and how it initiates that process. So first of all, we can look more closely at investigating the functional role of the GIH35 protein in understanding splicing quality control. Does GIH35 actively unwind RNA at any stage, or is it purely a marker for disassembly? So that's one of the questions that remain to be answered. Another question is, what is the RNA substitute or substrate of the GIH35 protein in the spBd complex? So that's another question that should be answered. We should also explore more closely the role of Gpl1 as a splicing regulator, right? Does Gpl1 participate in other splicing pathways, or is it exclusive to quality control, right? So we could look at RNA immunoprecipitation experiments or deleting Gpl1 in yeast to reveal how splicing fidelity and efficiency might be affected. Could artificially targeting Gpl1 to specific spliceosomes modulate the fidelity of splicing? That's another ongoing question that could be answered as well. So there's a lot of questions that remain with structural and biochemical characterization of alternative discard pathways. So for example, are there other structural variations of defective spliceosomes that are committed to disassembly? And how does the PRP43 protein helicase disassemble the spBd complex? That's still another question that needs to be answered, and so future experiments could be done in those areas. This research should help us learn more about understanding the link to human splicing errors and disease, but there's a lot more that can be done in that area too. For example, how do the human homologs function in mammalian spliceosome quality control? We've looked at bacteria, but now let's look at how this works in humans. And are mutations in these factors linked to splicing-related diseases as well? So we haven't really looked at mutations too much yet either. And lastly, discovering therapeutic or biotechnological applications. For example, can we develop small molecules that enhance the spliceosome quality control? And could artificial splicing sensors actually be engineered? And can we engineer biosensors that detect and report aberrant splicing in cells? So that could be an exciting area of research as well. So this was a fairly particularly interesting paper, and I kind of enjoyed reading this, and I kind of enjoyed, I really enjoyed looking at the Cryo-EM part as a structural biochemist. I was very interested in that, and I thought this was a great paper to look at that greatly enhances our knowledge of a very important process that's directly connected to the central dogma of molecular biology. And so I hope you enjoyed this video and learning a little bit something about the spliceosome and how it's involved in gene control, and how it's involved in the process of transcription and splicing out parts of the mRNA and preparing them for the ribosome. If you have any questions or comments or anything, leave them in the comments down below. If you like this video, go ahead and hit the like button, the thumbs up button on there. Go ahead. Go ahead and subscribe to this channel if you want to learn more about the scientific science and biochemistry on YouTube here. I've got lots of other videos, and I will see you again next week. Thank you. Thank you for watching Biochemistry Unfolded, from atoms to algorithms. If you enjoyed this content, please subscribe to this channel, and we will keep you informed of future episodes. Thank you.